So good morning, everyone. Thanks to those that have signed on remotely. We've got a group of about 10 here in person as well. So it's great to see this level of engagement for metaproteomics. This should be a fun week, hopefully informative. I expect lots of questions. That's usually the best way to figure out what you need to know. For future reference, we do hope to do this in the future as well, and potentially for proteomics as well. So we do want as much feedback as you can provide. So I wanted to just kick this off by thanking two participants, NSF. NSF funded the instrument that we're using for metaproteomics, and they funded it because of metaproteomics. And so this is an important topic to them, important enough to put an instrument in our lab, and we're supporting the workshop through that contribution. And second, Thermo, thank you for the snacks. So we'll probably have a Thermo rep later here in the week, so you can all say hello. But they are the instruments that we operate that you will become a little bit familiar with over the course of the week. And we're looking forward to introducing you to Thermo as well as Metaproteomics. So I now want to introduce Gustavo Diaz. Gustavo runs our proteomics services, has been heavily involved in metaproteomics since he's got here. Started off as a postdoc, now serving as a research scientist. And he will kick us off with a little bit of introductory material. Thank you, Gustavo.
I'm sharing the screen now.
One quick note, all of these materials will be available in the OneDrive that I have shared. If anybody online would like a copy, please send an email to Dorothea Lee. Cool.
So good morning, everyone. So what I'm going to do this morning is to give a
little bit of an echo.
No, sorry, that was me. Okay. I was just testing to make sure everything was good on the audio.
Just a brief overview, brief introduction of some concepts related to metaproteomics and proteomics in general, but most of them will be covered in depth with additional talks and when we are kind of having a tour for our instrumentation laboratory. So you have any questions, please let me know. So let me just set a pointer here. So at the beginning, I'm going to talk a little bit about the microbiome, which is the subject of the study of metaproteomics and metaproteomics overview. I want to give a brief overview of mass spectrometry-based proteomics, which is basically the same what we do with metaproteomics, but doing some steps additional because of the samples are very complex. And then I just want to highlight some challenges, but then there will be, as I just told you, covered by additional talks later the week. So what is the microbiome? This is the subject of a study in metaproteomics, and we refer to the microbiome as a community of microorganisms in a particular environment, place. We can talk about like a soil microbiome, if organisms living in a particular example of soil. And if we're talking about the human body, we refer to the microbiome as a community of organisms living in a particular anatomical localization. This is gut microbiome, skin microbiome, etc. So the study of microbiome has gained attention during the last, I would say, 3 decades. An increasingly large number of studies kind of showing that the interaction of the microbiome, alteration of the microbiome have heavy impact in human health and any other biological process in nature. And I just want to give you some ground numbers just to have a perspective of how important is the microbiome in the biosphere. This is here, our human body with all the living organs and the number of cells accounting for those is 10 to the 14th cells. And all of those are, like described from 23,000 genes. we overlay the number of invisible microbiome in all of our body, we are talking about 6 orders of magnitude higher number of cells and more than 9 million genes. So the interaction between these and our visible organs is the complete human, and alterations in this heavy number of living organisms is going to cause impacts in our health and any other, like a biological process. And a hallmark example of that is the atherosclerotic cardiovascular disease. Alterations in the gut microbiome can increase our risk of developing atherosclerotic cardiovascular disease. And why is that? So briefly, when we ingest high-rich protein content food, quaternary amines can be degraded by bacteria in our gut microbiome. If some bacteria are in higher abundance that are known as trimethylamine producers, TMA, which is a metabolite that is exclusively produced by bacteria, can cross and take our, like a portal circulation, get into the human liver, and for our metabolic machinery is transformed to TMA N oxide. And this compound has been associated with higher risk of development of cardiovascular disease. So having an alteration in our gut microbiome for methylated TMA producers is going to increase our risk of cardiovascular disease, whereas having demethylated organisms will have a protectant effect. So the effect of the alterations in gut microbiome is very extensive, it's comprehensive, and it goes beyond like a gut microbiome, which gut microbiome and diseases related to, like a food. But there is this, for example, gut-brain axis, in which alterations in our gut microbiome is being linked to mental disorders. And the evidence is that high that there are several clinical trials nowadays in which probiotics are used as in the intervention for mental health conditions, including depression and anxiety. So this is just a kind of key examples of how that gut microbiome can be, relevant if they get altered. If we go to soil, for example, just give you an example of the number of organisms in a gram of soil. It's impressive, the number of bacteria, 10 to the 8th, 10 to the 9th organisms in a gram of soil. And also, fungi, protozoa, nematodes. The number of living organisms in all those areas will modify or will alter all the processes that we have, for example, in crops, and just to give you an example. So to study this microbiome, the majority of approaches has been achieved via metagenomics. So metagenomics basically allow us to have an idea of the taxa that is in our microbiome sample. And at the same time, it provides information on the genetic potential of those organisms in our sample. But all of us know that the presence of a gene is not necessarily the presence of a messenger RNA, and definitely not a protein, a functional protein. So generally speaking, we can use marker gene studies to do these metagenomic studies, 16S RNA for bacteria, or 18S RNA for eukaryotic cells, or internal transcribed spacers if we are going to do fungi metagenomics. Or we can do just whole genome shotgun metaproteomics. And generally speaking, we go from reads to do some assembly reads to get context. Those contexts are grouped based on, like the probability of belong to the same taxa into potential biological taxon. And then we can also infer from those a functional annotation that give us the idea of what is the potential of this. But if we want to go beyond that, if we want to understand better what are those microorganisms doing, actually doing in a particular moment. A good approach is metaproteomics. We want to see proteins. I just want to give you this couple of seconds for you to go over this quote of Dr. Joshua Bayer about the importance of proteins in biology. And I will say that key word here is proteins are verbs, actions. We want to really know what are microorganisms doing in a particular moment. We really want to see what proteins are being produced. The key example here is enzymes. Like every single metabolic process that we see is achieved via the participation of enzymes, proteins. So knowing what proteins are being produced in a particular sample, we're going to have a better idea of what was the actions, what was the responses that were being generated by those organisms in our microbiome sample. So the study of those proteins in those communities is metaproteomics. This is the definition that was coined 20 years ago as the large-scale characterization of the entire protein complement of environmental microbiota at a given point in time. And the study of these, the proteins in these communities, allow us a direct link to phenotypes on a molecular level. What is the phenotype? It's key for us to understand what is happening. Also show us the adaptation of those organisms to experimental or natural cues, insults. And it's definitely a great complement for the other omics. So we get from metagenomics a real survey of the taxa composition. We know what is the potential, functional potential. We get from metatranscriptomics the idea of what potentially happened. But you know that transcripts are not proteins. They are heavily regulated. But the fact that we have proteins is a very strong evidence that the situation, the process was actually happening at that particular time under that particular conditions. So when we study metaproteomics using mass spectrometry, the workflow is very similar to the workflow that we use for single organism proteomics. When I say single, it can be like 1, 2, or 3 organisms that are normal, like a normal shotgun proteomics approach. In metaproteomics, we are studying the whole community. But the process is overall very similar. We start with sample, we extract proteins, we digest proteins using proteases into peptides. Peptides are analyzed in a first mass spectrometry step, we call the MS1. We infer from this the M/C of those peptides and the intensity, and this is a surrogate for abundance, for relative quantitation. And then some of those peptides are selected for fragmentation, and the fragments generated are analyzed in a second mass spectrometry step, the MS2. And the analysis of these fragments allow us to have an idea of the amino acid sequence. With those sequences, we get protein identities. So at the end, we end up with a list of protein abundances and protein identities. I will go deep inside this in further talks. So basically, what we have is our mixture of peptides that are separated by hydrophobicity using liquid chromatography, and then they are ionized to be able to be, analyzed inside the mass spectrometer. And we generate this. This is an example of a chromatogram. See, some of you will be familiar. It's just M/C's, intensities over time. All those ions separated in time by hydrophobicity. And then we have the MS1 that I was just mentioned, intact peptides that are used to infer protein intensity, concentration or quantitation, relative quantitation, and then the MS2 that is used to infer amino acid sequence. So the process for protein identification, very generally speaking, what we do is like using FASTA files, like a sequence obtained amino acid FASTA files, We generate in silico using algorithms and computer programs all the potential, all the theoretical peptide fragments, and then the fragments of those peptides, all the possibilities with different charges, different modifications, and then doing an exercise, an algorithm of matching the raw data with the experimental data. Some thresholds are applied, and some of them are selected to give us the information of peptide sequence and for the protein identification. When we see the workflow applied to metaproteomics, it's a little bit more complex. And this is a great work for you to review. This was an exercise known as the Critical Assessment of Metaproteome Investigation, CAMPI. And this is an initiative to put several laboratories all around the world in the consensus to have comparable results. So this is very key because there are so many variables when we do metaproteomics that could affect the results and the analysis. The first one, challenges with sample, and Dora will go over this in more detail. So we need to account for the variety of cell types that we have: gram-positive bacteria, gram-negative bacteria, cells that are with a strong cell wall, actinomycetes, fungi. So we need to apply a combination of either mechanical or chemical and mechanical cell disruption to get proteins out. Metaproteomic samples are very complex. There are several. I mean, in a normal sample, we have a very complex mixture of proteins. In a metaproteomic samples, it's even higher. So fractionation is very important. At the peptide level, at the protein level, we need to be mindful on that. And what is the strategy applied? How be consistent with that? And then at the bioinformatics level, that's a super key aspect that is still under development. Because as you know, like when we have human proteomics, so we have the FASTA file of the human proteome, easily available in UniProt. And we can do all the in silico predictions. And when we have metaproteomics, it's a big problem at that level, at the level of what is the information we're going to use to match our raw data. So first thing is, there will be a bunch of missing information. There are so many organisms that are you cannot put into culture. Ergo, it's highly probable that you don't have genetic information of those. So there's de novo approaches that are super challenging, and they are still not well developed, especially for metaproteomics. So we still rely on having a FASTA file to do the matching. The best possible scenario is for all of us is to have a metagenome-derived proteome, metaproteome. That will make your life and our life easier and more, like the more accurate results. We use a metagenome-derived information to do the analysis. This is ideal. If not, we need to rely on public repositories. Or let's say we have this idea of using the pan proteomes that we have available from, for example, all prokaryotes. This is just an example of when we have Bacillus subtilis. We're talking about 4,000 protein entries. And we take all the prokaryotes, all the proteins that are available. We are talking about 36 million entries. When we do the search against 36 million entries, the all possibilities increases our likelihood of false discoveries by a ton. So unreliable results, variable results, difficult to assign results. So these are important considerations. I just want to mention one approach. There are several approaches to deal with this challenge of the not having a metagenome-derived proteome and having to use public repositories. And the rationale is using a two-step approach. One step that allows us to decrease the search space, and in a second step, using stringent filters like very low false discovery rates, and doing the actual identification. This is one of the approaches. We will be using this, we will be applying this approach that I'm referring in this slide here, that was developed by Dr. Pratik Jagtap, collaborator of us from University of Minnesota. And this is just using the first step without any fault discovery rate filtering. And then all the hits that we get, are used to develop a smaller data set. Just to show you how this improves the number of identifications, this first database here has over 6 million entries. We use this one to do the analysis. The number of identification, the identified peptides, is around 5,000. Applying two-step approach, we use this first without any FTR filtration, and then a subset of those proteins that were here are selected to build this one that contains only 9,000 proteins. The number of IDs dramatically increased. So we have a gain in identifications that is important. So we'll be just going over this strategy in our sessions of Wednesday and Thursday. But just to show you how important it is to decrease the size of the search space. But this is only proteomics, like basic proteomics. We are trying to assign raw data to proteins, which is important, which is key. But the real kind of gain in metaproteomics is the functional analysis. And to have an idea of functional analysis, we need to do some taxonomic assignments using peptides. So that's the two additional gains when we do metaproteomics. How we get from a list of peptides, assignment of those to taxa. And that's super difficult because of this concept of peptide degeneracy, which is one peptide can be linked to 200 taxa. Which one is the correct one? It's difficult to know. Sometimes the algorithms won't be able to manage that, but there are some approaches. And the other one, perhaps in my opinion, the most important one, is how we do an assignment of function based on peptides. Again here, you know that a particular peptide can be linked to 100 proteins. If we do that with a human proteome, there are some peptides that are shared for several proteins. The assignment of that particular peptide to a protein is difficult in a single organism. Having a taxa, a combination of taxa, an entire community is much more critical. So there are some computational approaches to do that. And we have invited Dr. Tim van de Buch. His group has developed a lot of informatic strategies to help us to do these connections, peptide function and peptide taxa. So this is going to be our talk on Wednesday. So please be here just to see how we deal with this. So I think that's all that I have for today, this quick introduction. So if you have any question, please let me know. So questions? Yes, Laura?
So I know this is going to vary against, what people have looked for, what people have done metaproteomics or metagenomics for, and basically what people are interested in studying as to like how the search space for what's there is even available. I'm just wondering, I've started a thing with algae. It's the metagenomic space is poor as far as like 18S annotation for algae. You have any sense of whether or not there's any annotation for algae in proteins? Is that a thing of interest to anyone?
Yeah. So I'm going to say your question.
Yeah.
So Lara is asking about availability of protein annotations, protein sequences for algae. Yeah, I'm not very familiar. We haven't received studies on that regard. We have some like C microbiome studies. We have some, like a metagenome derived information And with that, I can tell you just one example. You work with an algae, right? So, yeah, or is it, and when we have the FASTA file derived from the sample, we get like a beautiful proteomics and identification. When I say beautiful, it's like 2,000, 3,000 proteins, which is impressive. When we don't have that, normally the fault discovery rates, it's everything out. I'm not familiar with the number of protons that we have available for based on 18S RNA. Yeah, but there will be uniprot sequences that we can use. Yeah, but what I see was like, for example, if we have repositories for metagenomic information, even in different geographical localizations. Just to give you an example, soil microbiome. We have information available from studies that were performed, different states, different soil characteristics. I use that to do identification of proteins in soils from Colorado. And the ID rates were high, were great. Comparing that to metagenome-derived data from the sample that was under study. So it was good. Versus, if we compare that to general repositories, general protocols. So I think like the idea of having a metagenome-derived information is key. Still, you will see that bioinformatic approaches can help us to deal with that, even if we don't have that information.
Thanks, Gustavo.